Alright, I'm going to recap you on the lab that you were supposed to be doing in class today. You are only going to have a few moments in class tomorrow at the beginning of the period to finish up with any of the pop beats. So where everybody seemed to be getting messed up is with the restriction enzymes. So the extraction part, no problem. We can set up and we can make our DNA strands, copying the order where A's are blue, T's are orange, C's are yellow, and G's are green. Okay. So the strand that I have on the screen on the right hand side is the crime scene strand. Okay, so the first one, again, the one we did in class, I need you to do suspect 1, 2, and 3 with your partner or your group or on your own. So if I scroll down in the instructions a little bit, and I'm looking at the restriction enzymes, which are here, we have two separate restriction enzymes, enzyme 1 and enzyme 2. Enzyme 1 wants me to find a segment of DNA in my strand that reads ATTA. If it is ATTA, that means it is going to be blue, orange, orange, blue. So the trick here is to look for double orange. And if I find two oranges, if there are blues on either side, then I'm at the right segment of DNA. So if I scan through my DNA, oh wait, there's two oranges. Okay? And I have blues on both sides of those oranges. So with it being a restriction enzyme, and restriction enzymes acting as DNA scissors, cutting it, I'm able to break apart that portion of the DNA. That's enzyme 1, restriction enzyme 1. I have one cut. Now I have to go and look for enzyme number 2, which is CGGC, or yellow, green, green, yellow. I have to go back through the entire strand of DNA and see if there are any places where that restriction enzyme will work. So as I travel through and look at the strand, oh wait, there are two greens, and on either side of that green are yellows, so that my restriction enzyme 2 will work here. So I have another cut with my DNA scissors. Keep going through. Oh wait, there's another two green with yellows on either side. So my DNA scissors restriction enzymes also works here. Now I have 4 separate pieces of DNA. 1, 2, 3, 4. We talked about how DNA fragments move through a gel in gel electrophoresis based on size. I mentioned sand and rocks in a colander and which one would come out first. You told me the sand because the sand is smaller. Same concept applies with the DNA fragments. So if I take all of my fragments here, if I keep the prong on the right hand side because that's the way it was facing, they're all in the right order. I'm going to place my fragments in order from largest to smallest based on the number of beads. So I have one that has 8, another one that has 7, one that has 6, and one that has 5. So largest to smallest. I'm going to turn the paper. So if I look at my grid, I have crime scene, suspect 1, 2, and 3. If I look at the rows, I have numbers, 10 through 1. Obviously, you know that 10 is the largest, 1 is the smallest. Think of it like a funnel. If I have a funnel and I have strands or segments of stuff, and say I can fit 7 across this area. Those 7, if I want to keep them together, they cannot go any further because they will not fit. So as I move further down into my triangle, I have fewer and fewer beads in my strands based on what can fit. Okay? So I'm decreasing in size as I go down. So based on the number of beads in your strand, you place that in the row that has that number of beads. So the very first one, the largest piece of DNA that we have was 8. So I'm going to take that strand that had 8, which is this one, and I'm going to color code it into those boxes. Or I'm going to make a bead strand. So I have green, yellow, orange, blue, yellow, yellow, orange. Okay. 1, 2, 3, 4, 5, 6, 7. Oop, I messed up. This is supposed to be blue, and this one's supposed to be orange. See? 1, 2, 3, 4, 5, 6, 7, 8. Okay, good to go. I'm done with that fragment. The next fragment had 7 pieces. I color code those 7 into the 7 row. Orange gives me 1. Blue 2. I have 3 greens in a row. And then a yellow. And then a green. Oops. So I have 1, 2, 3, 4, 5, 6, 7. Checks out. Okay, I'm done with that fragment. My next fragment has 6. So in this the row for 6, blue, orange, blue, yellow, yellow, green. 1, 2, 3, 4, 5, 6. I'm done with that fragment. And your last one, you have 5, which would be green, yellow, orange, blue, yellow. 1, 2, 3, 4, 5. I'm done with that fragment. Those are my 4 fragments for the crime scene. Now you have to do the same thing with the DNA samples from suspect 1, 2, and 3. When you use your restriction enzymes, because it is different DNA, you may not have 4 pieces. You may have 3. You may have a fragment that only has 2 beads instead of 5 being your smallest. That's where you have to look at the specifics within your restriction enzymes. Remember, you have to use both enzyme 1 and enzyme 2 to cut your DNA strands. Once you have filled in the gel electrophoresis page, the front page with the color coding, all you're doing from that point is you are comparing the 3 suspects against the crime scene to figure out who the guilty party is. Once you figure out the guilty party, you can answer your questions. What are the bands and why are they important? Which suspect is the murderer? Explain how you got to that answer. I basically just did that for you. Possible controversial aspects, meaning, can this be 100%? Is there a chance it could be different? Am I wrong? And then how might DNA fingerprinting be used to determine if a child belonged to specific parents? We've done an example like this. So think about what we've talked about in class. And then you have 2 practice ones, which technically we've already done because you already did those 2 practice ones in class earlier today. So see if you can do it again. Hopefully that helps it make more sense. Again, this really was not meant to be as challenging as many of you were making it. Okay? Just take it slow and look for similarities. Hopefully that helps.